chat with you a little bit about the. I want to move over to certainly gene therapy. I want to talk through the strategies for development for the next generation AAV capsids. So, can you talk us through what some of those strategies, in fact, what they are?
Sure. Um, so, right now, there are many AAVs that have been discovered, sequenced that are naturally occurring, and a small handful of those have actually made it into the gene therapy realm and been used in programs that are going into the clinic. The ones that have floated to the top and are most popular, certainly work. They have targeting characteristics that can treat disease. But I think the field is now recognizing that the naturally occurring AAVs have both strengths and drawbacks. So a good example of this is AAV9. AAV9 works really well to transduce certain cells and can be very effective. But one of the drawbacks is that if you give it systemically, a huge amount of it goes to the liver, and that can cause significant toxicity in the patients and adverse events. There are other AAVs that have been used in different programs, and all of them sort of have that same drawback. So a lot of companies are now focusing on developing next generation capsids that enhance the strengths of the currently existing AAVs and potentially ameliorate some of the risks and the weaknesses. I think the major focus around developing the next generation AAVs is on targeting, trying to develop capsids that specifically go to the place that you want them to go when they're introduced into the patient, and on manufacturing. So developing capsids that are easier to manufacture and provide higher quality drug product with fewer contaminants and empty capsids and things like that. So there are a couple of different strategies that have been explored for developing these next-gen capsids. In the early days, people did a lot of random mutagenesis. So you'd build a library of 10,000 different capsid proteins that had amino acid changes sort of randomly sprinkled throughout the protein. The problem with that is that the capsid itself is a very complex structure that's composed of about 60 of these proteins that are virtually identical, sort of all associated together to encapsulate a viral genome. If you change the protein structure of the capsid protein in very small ways, that structure can just completely fall apart. So if you use random mutagenesis to construct a library of 10,000 constituents, you may find that less than 10% of those will actually assemble into a viable capsid. So there's a lot of work creating the library and a lot of work trying to manufacture things, and then you end up with a relatively small handful of capsids that you can actually put into animals and screen. So people have tried to come up with better ways of generating these libraries of different capsids to be able to screen. The company that I worked with before AVROBIO, it was called Affinia Therapeutics, and their platform is based on science that came from Luc Vandenberg at Massachusetts Eye and Ear. And his strategy was to take to make a phylogenetic map of all of the currently existing AAV capsid proteins, group them into clades and sort of understand how the various AAVs that are existing in nature are genetically and evolutionarily related, and then walk back through evolutionary history, trying to predict what the sequence of their evolutionary ancestors might have been, on the assumption that if an AAV capsid had existed at some point in evolutionary history, then it must be able to assemble into a viable capsid. And so using that approach, he was able to build libraries of a very large number of capsid proteins with various changes to the protein sequence. And it was very successful because most of those capsids will actually assemble into viable viruses that can package a genome and deliver it to cells. So Affinia is screening those libraries, looking for unique manufacturing and unique targeting characteristics. Other companies are doing similar things where they're using a more refined targeted mutagenesis approach in areas where they think the amino acid changes are less likely to disrupt the assembly of the capsid. And then other companies are also working on taking existing AAVs and appending small groups of amino acids to specific areas of the capsid protein in order to increase cell penetration or promote targeting to certain tissues or certain areas within the patient's body. So, all of these things are in the very early days, but I think one of the key things that's coming out of this, aside from the fact that you might discover a capsid that works really well for a specific disease indication, is that by doing this kind of analysis of how different changes to the capsid proteins affect targeting and behavior of the capsid, we're learning a lot about AAV capsid biology. Where can we make changes without disrupting the assembly of the viral capsid? Where can we make changes that actually affect targeting in different tissues? And as we understand that more and more, the generation of libraries and the screening techniques are going to become more and more refined. So it's an exciting time in AAV biology because we're investing so much money in experiments that really help us understand the nature of all of the different areas of that capsid protein and how it affects the biology of the virus as a whole.
Okay.